Hello everyone, welcome back to my channel Bani's Biology. Today we are going to break down one of the most elegant idea in molecular biology, the Wobble Hypothesis. But before we get through, let's quickly walk through the journey of genetic information. It all begins with transcription, where DNA is copied into mRNA, and then the ribosome, this is the ribosome, bind to the mRNA, and during translation the cell reads the message in group of 3 nucleotides and each triple forming a codon, and these codon form the genetic code. So we all know that in the genetic code there are 64 possible codon and 3 of the 64 act as a stop codon. So these are the stop codon UAA, UAG and UGA, and the remaining 61 codon, also called sense codon, encode the 20 amino acid. For translation, to decode these 61 codons require a tRNA molecules with an anticodon with which it can stably complement. So if you look at the structure of the tRNA molecules, this is the tRNA structure. So you will notice it is a small RNA molecules, and this end is the 5' end and this end is the 3' end, where the amino acid attached. This is the amino acid attached site, and down here have the anticodon, a 3 base sequence. So this is a 3 base sequence, and this anticodon pair with the mRNA codon. This is the mRNA codon through base pairing rules. The classic Watson-Crick pairing of A pair with U and G pair with C. But here the twist, Francis Crick realized that if each tRNA molecule is paired with complementary mRNA codon using Watson-Crick base pairing, then 61 types of tRNA molecule would be required, since most organisms have fewer than 45 types of tRNA. So Francis Crick proposed the brilliant idea in 1966 that the third base of the codon is flexible. It can wobble. So let's take an example to understand wobble. Suppose the codon on the mRNA is 5' AAC 3'. This is the mRNA codon, and this is the first base, this is the second base, and this is the third base. And its complementary anticodon on the tRNA would be 3' UUG 5'. This is the anticodon on tRNA. So this is the first base, this is the second base, and this is the third base. Count from 5' to 3', and this is the complementary base. So now look carefully at the third base of the codon, the C in the AAC. This third position is special. It is the wobble position. This base can sometimes pair with the more than one nucleotide on the anticodon, and these first two bases of the codon always follow strict Watson-Crick base pairing. They are stable and called standard bases, and also ribosome hold the first two bases tightly, but the third bases of the codon and the first base of the anticodon, this is the first base of the anticodon, are more flexible, and this base can form non-standard base pairing and allowing one tRNA to recognize more than one codon, because this position can wobble, and Francis Crick called it the wobble bases, and the idea is known as Wobble Hypothesis. So this image displays two identical tRNA, both carrying same amino acid leucine, and they have the same anticodon GAG. So according to Watson-Crick base pairing, normally anticodon, this is the anticodon GAG pairing with codon CUC. So it is the Watson-Crick base pairing. But here wobble pairing, the same anticodon GAG pairing with codon CUU instead of CUC. So the third base of the codon, this is the third base, that is U, is flexible, and this flexibility allow non-standard base pairing. So G in the anticodon normally pair with C, but G can also pair with U at the wobble position. So one tRNA can read multiple codon. This is the same tRNA can read the CUC, also CUU, So multiple codon can recognize. So this flexibility at the 5' anticodon position, so this is the 5' anticodon position, can make small movement and that adjusts the geometry of pairing. And this wobble pairing allow single tRNA can interpret multiple codon and making translation far more efficient. So this wobble pairing can allow one tRNA can read multiple codon. But only a few specific non-standard pair are allowed. Like G can pair with G in the 5' end of anticodon can pair with U or C. This is a 3' end of codon. So this is the 5', like example A, G, A. 3', this is the codon. Next is the 3', U, C, U. 5', this is the anticodon. So this is the G in the 5' end. This is the 5' end of anticodon. Can pair with the 3' end of the codon. This is called 3' end of the codon. U or C. Next C can pair with G. A can pair with U. And U can pair with A or G. And inosine can pair with A, U or C. So this limited combination are called Wobble's rule. But why is wobble pairing restricted to only certain base pairing? Because this wobble pairing maintain a ribose distance. So this is the anticodon and this is the codon. So this wobble pairing maintain a ribose distance close to that of normal AU or GC or Crick base pairing and keep the overall geometry of the codon-anticodon helix acceptable for the ribosome. But not all bases can pair in the wobble position. So if purine pair with the purine, this pair are too wide. So you can see this distance between ribose that is too wide and the ribose distance become too large. And if pyrimidine pairs, that is U or C or U, are too narrow. So this is the narrow and the distance become too small. And both of these distortion would disrupt the geometry of the helix. So here the structure had purine pairing. Here the ribose distance become too large. So that's why wobble pairing is limited to only a few combination and allowing fewer tRNA to decode all 61 codons. So in conclusion, the wobble hypothesis explains how the cell maintain accuracy while still using fewer tRNAs. So the first two bases of the codon pair strictly through Orson rules, while the third basis is flexible and allow limited non-standard pairing. And this control flexibility keeps the geometry of the codon-anticodon helix acceptable for the ribosome and allow one tRNA to read multiple codons. So if this lecture boosted your understanding, don't forget to like, share and subscribe for more lessons.